Let's look into another enzyme that is commonly used in genetic engineering, and this enzyme it is called as reverse transcriptase. You have known about transcriptions, which means it is where the DNA transcribed into mRNA. So inside a nucleus, you will have transcriptions activity. Reverse transcriptions means reverse of the process. Instead of DNA making into mRNA, mRNA can be made into DNA through artificial process inside the lab. So it is going to convert it into DNA, and this is called as reverse transcription. You require enzyme in these reactions, and that enzyme we call it as a reverse transcriptase. And the DNA form here, since this is the one that produced from the mRNA, we don't simply call it as a DNA, we call it as a complementary DNA or cDNA that is complement to the mRNA here. This enzyme, reverse transcriptase, you can find it naturally in retroviruses. Now, retrovirus is RNA virus. So inside these viruses, you will have nucleic acid that is made of RNA. So this is the RNA inside. So when this virus going to invade into the host cells commonly they have a DNA in their nucleus. So they can't incorporate the RNA directly into the DNA genome of the host cells. They need to have an enzyme that is called as reverse transcriptase that going to help them convert their RNA into DNA before incorporated into host cells. And this one, reverse transcriptase, can help to produce from the RNA into a cDNA that is single-stranded. Let's look into the process of reverse transcriptase. To do this, it is one of the methods where the scientists can be extract the mRNA out from cells and then converted into cDNA and then incorporated or do the gene cloning. The process is always start with mRNA extractions. That is the animal cells, and you have a nucleus inside here, the biggest organelles in the cell. Outside of the cell, outside of the nucleus membrane, you've got cytoplasm. In the cytoplasm, you've got a lot of this single-strand mRNA that is produced in the nucleus and being transported out from the nucleus into the cytoplasm. The first step is extraction. So extract the mRNA out from the cells. This is a test tube. After your extractions, you have a lot of mRNA, single-stranded strand. All mRNA are single-stranded. Since you already extract all the mRNA inside here, add inside this test tube with reverse transcriptase enzyme. The functions of reverse transcriptase is to convert this mRNA strand into cDNA. So they start to screen each of these mRNA strands and they produce a complementary DNA. When you have a cDNA, your purpose is to want to have clone DNA or transfer the DNA to another vectors or another organism. So you need to clean out all the mRNA. So the next step will be remove mRNA. Now inside this test tube, you will have solely of all the cDNA inside without any mRNA. Notice that both of these mRNA and cDNA, they are single-stranded, meaning it's only one, like it's not double-stranded. For cloning, you can't clone single-stranded because all your vector plasmid are double-stranded and all your genome are double-stranded. So in order to make it same double-stranded, you need to have extra step here, which is add with DNA polymerase. Put the DNA polymerase inside. The functions of DNA polymerase is we learn about DNA replications. It is going to replicate the DNA. Same thing inside here. All these single-stranded cDNA now will become double-stranded cDNA. This is essential for cloning purpose. And the next thing that you can do is this amount is not really enough for successful cloning. To multiply it to millions copy, you will need to have PCR machine to multiply the DNA inside. So you will have many of these DNA inside these solutions. Could be up to millions copy. And this is the process of reverse transcriptions. Reverse transcriptase enzyme is involved in reverse transcriptions. The process of reverse transcriptions is looking something like this, where you isolate the mRNA from the cells, mix it with reverse transcriptase enzyme, and it will form the complementary cDNA. Remove the mRNA, add it with DNA polymerase, and the double-stranded cDNA is produced by semi-conservative replications by the help of DNA polymerase here. And the last step is you can follow by PCR or polymerase chain reactions to mass produce the cDNA up to million copies before cloning or before gene transfer activity.